Thank you so much. I'm excited to be here. I believe that Profound is one of a success story in Canada. We have done a lot in short period of time. So today my goal will be to, in 1 hour, share what those critical learning that we had. Um, and I believe that, you know, learning that we had can help you if you are in that area of innovation and commercialization. So hopefully I can speak to your needs. There is a lot. I'm trying to cover little bit high level on TULSA, what TULSA is, and the technology that we have developed. Then a little bit on a clinical strategy. What did we do for clinical strategy and regulatory strategy? And then I will end with any Q&A. Hopefully we have 1 hour and some time for Q&A, and I will cover all that. So we can start with TULSA-PRO. As Ahmed mentioned, it is a system that is designed to ablate prostate. So given any innovation, you look for why you need to bring that to commercial world. So you go look for an unmet need. And for us, unmet need was that prostate cancer treatment had options. It's not something that cannot be treated. So we are in the area where you knew that there are alternative options. What your technology was designed for is to have a better quality of life. So that's where we started. It's like, okay, there are other treatments, but we can do better. The technology itself was unique. There is nothing like that where you use planar energy inside the organ and you can do a certain distance from the organ. So the transurethral directional ultrasound, a unique way to ablate tissue. I'm trying to only give you a little bit, so when I talk about clinical strategy and regulatory strategy, you can connect with these points. And it is combination of ultrasound in MR, which is what a scientific uncertainty, brought scientific uncertainty and engineering challenges and regulatory challenges. How do you do that? And then it is not usually ablations are like a point-to-point ablations, but this was like a sweep. So you put something inside the body, you do the turn, and it does the trick. So those were the main things that we started with when we started doing our strategy. From a value proposition perspective, TULSA was non-invasive. So for patients, it's like, it's a benefit for them. They don't have to deal with the heal up time. And it is giving a cooling methods. So when you're heating someone, you will burn good and bad tissues. So the cooling method is protecting critical structure. So that was a value. And it could heat from inside the body. Usually HIFU treatments are from outside. You are focusing inside the body. This was unique in that sense. And the real-time MR planning is, you can imagine the technical challenges where you are sending high RF energy inside the MR and hoping your system stays quiet. So it can give you good images, and those images then can be used to plan the treatment and then execute the treatment. So these are the challenges that we started with. A little cartoon here showing how it is done. So patient gets in the hospital, gets through a planning phase, and they are brought into the MR room. Device is inserted in the patient, and then the MR imaging starts and the treatment starts. So what you see here is image where you have number of slices, and each slice is aligned with the transducer. So you have a planning images, and the transducer is emitting heat to that plan. And the boundary that you see here is where the algorithm was developed to stop the heat at the boundary. Sounds very simple. This is a real patient treatment. I will walk through the challenges that we have. The device comes out, patient comes out. Our trial have shown that most patients went home same day. Some stayed overnight due to the timing, but the treatment was, from a morbidity perspective, it was like patient recover from general anesthesia and they walk out from the hospital. So this is what we started with. I'm just giving you a little bit overview of what the system looks like today. And there are components that are inside the MR room, and there are components that are outside the MR room. So inside the MR room is MR compatible ultrasound applicator, which you see on a right-hand side. It's a sterile device. And then there is a rectal device, which called endorectal cooling device. This is non-sterile. These two devices are disposable. So business case, when you're talking about how you design your system, there were elements went into this to say, how does it support the business model? So this is a recurring revenue business model. And then all these are coming together in a workstation, which is the operator's workstation. You can do the acquisition, planning, and execution of the treatment. So that's the overall view of the TULSA. Clinical workflow is, as I mentioned, patient comes in, gets prepared, go under general anesthesia or sedation, device gets inserted, patient gets in the MR, images started, as soon as the images are in our system. So we are getting images from the MR to our system, doing the planning there. Once the planning is done, system communicates with MR on a continuous basis. So every 6 seconds, we get images from MR, and those images are used by robotic arm, and it calculates the frequency and power to see what we need to ablate. And it is a closed loop system. Once that treatment starts, MRI sending the thermometry images, which our algorithm uses to stop the heating at the boundary. So that's what real-time MR and the unique temperature that we achieve. Once the delivery is done, we have confirmation. So you can do the contrast enhanced imaging to see what non-perfused volume do you have. So it's right after the treatment. And these images are coming from our trial. So that was just a little bit of 10 years of work. But just to give you an idea of what this technology is all about. Now you have a concept that was born here at Sunnybrook, and you need to commercialize it. What do you do? There are major components. Of course, you need money. If it is a medical device, you need a clinical data. If it is a new technology, of course, you need a clinical data. And then you need to have a design development strategies for commercialization. So regulatory strategy is not something that you can do independently. You need to work with those three elements and come up with how do you really take from one stage to the next stage. When I say good enough, yes, it is subjective, but there are decision made during that development process, right? Have we answered all the scientific uncertainties? Do we have a working prototype? Is it good enough to put it in animal? Is it good enough for human, right? These are major decisions. And regulatory can design things with clinical team, but they cannot do without knowing how engineering is working, what challenges they have. So once you learn that, that's when you go back to regulator if you need to. Most of the time in my career, I have taken advantage of talking to regulators. And you can bring your questions to them and validate what you're doing is gonna get you to the approval stage. I'll touch on all those things. Now I will just show you, I put here 2000 because that's what I'm aware of as early 2000 is when Sunnybrook has this concept. I believe it was, I know it was Dr. Rajiv Chopra and Dr. Michael Bronskill who had this project with Dr. Klotz. Very early, they had an idea of sending this ultrasound energy in the MR and watching it in a real time. And it started early 2000, and today in 2019, we have this product in the market. It may sound discouraging that it takes this long to develop something and commercialize something. I would say no. But I'll walk you through what happened in those periods. So this is the very first system that was at Sunnybrook. Then the system was evolved to do a preclinical work. This system was used on animals. Then we modified the system. So in 2008, Sunnybrook did 8 canine study, and that's when the company was formed. So in 2008, Profound was established, and we had this system to do preclinical work. And preclinical work is where, when I say good enough, is we took what was ready from a product perspective, from a prototype perspective, and we went to FDA with our questions and say, we are going to design a preclinical study. What would it be? And would it support my clinical trial? And there were a number of questions asked. And based on those questions, we were able to design phase 1, and phase 1 was done with the system that you see. Today, the system looks like this. So the engineering challenges are, especially for this technology, has been significant, just because high power and high energy and high frequencies that you use within the MR, it's not easy. It's like keeping the system quiet and making it work so treatment can be done without interruptions. And you are relying on MR technology, which has other challenges. You are relying on other companies' work and collaborating with them ahead of time to develop the interfaces that you need to develop. So I love, this is one of our scientists who drew this one. And it is the reality for us. So every challenge that you have, we worked backwards. So this really reflects how did we answer many, many questions. So if you have so many things going, you have development going, you have scientific uncertainties, you have small team, you have little money, you have to develop a strategy for commercialization, all those could be major projects on its own. But at some point, you have to decide which one is your priority and when. So decide what is your end goal and take each end goal, work backwards, right? And then you ask, all the time you ask question, what are we trying to answer? So you always look for the question that you're trying to answer. And that's how you create the focus. And then time is like, do I have 10 years to develop this? Like every project that we had was a very sensitive, had a very sensitive timelines to it. So we watched very carefully each project, how do we deliver that? And then of course, each project needed some money. When I started with the company, company only had $9 million. That was in 2011. So in $9 million, we were given number of tasks to achieve, right? And we did it. And to do something like that, you need to have clarity, and you need to have focus, and you need to have alignment. So if every department working on something, you need to at some point come together and see if your strategies are aligned or not, your projects are aligned or not, your sub-projects are aligned or not. So that alignment is what got us where we are today. I'm going to talk about clinical strategy. So going back when we started this project, it was, there were a lot of questions. Is it a cancer treatment? Is it whole gland treatment? At that time, HIFU was a buzzword, everyone talking about focal treatment. So do we want focal treatment? Do we want BPH? Forget about cancer. Can we just do BPH? It's faster. It can be done in less money, huge market. So you can take any of this and create a whole new project if you want to. And that's what we had as a challenge. As you have options, which one do you pick? On top of that, this is a Sunnybrook study. There were key opinion leaders were telling us that TULSA is perfect for focal. If you know about HIFU, you are sending energy from the rectum to prostate, and it has its challenges. where TULSA didn't have those challenges because you are not sending energy through other organs. And it was very convincing. Dr. Klotz from Sunnybrook did this study with Dr. Rajiv Chopra. They identified the tumor and they ablated the tumor. This was a treat and resect study, excellent results. But was it good enough for commercialization? Right? So you have these options in front of you and you have to make a decision. Which one do you pick? So after, our team, and I had Matthew Burtnick, who was from Sunnybrook. He did his PhD on this topic. He joined Profound, and he was by my side through this journey. He was a scientist for this technology. And our initial approach was talk to urologists and radiologists who are key opinion leaders in the area and get their feedback. Get to know what would they do if these are the options. And we decided, talking to the key opinion leaders and regulators, that our device is really good for quality of life, and we need to treat the whole gland because there was diagnostic issues that with focal, the biggest issue was that if you cannot identify where the tumor is, you cannot ablate where the tumor is. And if you have a multifocal disease and you cannot see it in MRI, you will miss the tumor. So we decided to go for whole gland, and we said that we want to do it safely. So our value proposition was that we will do precise treatment and protect the adjacent organs. And that is how we decided our clinical strategy. Less morbidity was interesting for prostate cancer patients. So we did talk to cancer patients, and for them, surgery was, it's like, yes, I can treat my cancer, but I am afraid that I will put diaper for the rest of my life, right? So we heard them, and we felt that morbidity, if we get a really good results, this technology will survive in a commercial world. So this is what the phase 1 study was when and the very first study that Profound conducted. And this study was done, again, there is a number of things going on in this slide. We had limited money, and we wanted fastest path to regulatory approval. And to do that, we knew that we will not get that in the US because we talked to regulators and we understood. And we decided to go with a European approval. We talked to notified body and we had a very good idea what they are looking for. So with this study, we created a protocol where our goal was to treat low risk to intermediate risk patients, which will show a good disease control and a good, quality of life questionnaire, quality of life profile. When we submitted this protocol, we was not allowed by US and Europe, German BfArM regulator, to include intermediate risk patients, but Canada did. So what we did is we increased our enrollment in Canada. So we treated more in Canada, we treated second in Germany, and then least in US. We knew that we have to go to US after Europe. So we knew that we needed to do another study there. So if you just did one study and you thought you would use that study for US, Europe, and Canada, you will fall into this controversy where most people said that prostate cancer, low risk, doesn't need treatment. So even if you show a great quality of life profile, but people will say, why to treat patients who don't need treatment, right? So you don't have a commercial success. And once you treated those low risk patients with the exploratory endpoint of cancer, we were showing the results that are interested for people who are treating cancer or people who want the treatment for the cancer. So the study was designed in a way that one country will get us approval, one country will get us a larger population exposure. Patient or doctors will be able to see what we have achieved with this study, 30 patient study. So that's kind of a strategy went into this. This is not answering all questions, but this will be answering major questions that we started with. So you have study design, you have an idea of, what needs to be done, but then you need to know how it needs to be done. Who will do this? And as a very small group, it was only myself, Matthew, and another person, so 3 people in the department. And we had to decide if we have to get CRO involved, or should we do it ourselves. As a small company, when we got quotes from CRO, we said, no, we'll do it ourselves because it was very, very expensive. And none of us had an experience in clinical trials. Myself, I had a regulatory experience, but I have never done trials. So it was a risk that I mess up. But I was confident that, if I understand the environment, I will involve subject matter expert and I can do it. So I asked my CEO if he will allow me to do it. I presented the quote and I said, this is what it's gonna cost. Either we write a check for that or we trust me that I can do it. So this is important for people to understand. It needs certain skills for sure. But if you are in innovation and if you don't have enough money, if you want to collect clinical data, you need to understand what those options are. And the key factor for clinical trial is not protocol itself, it's the decisions that I was talking before. Those decisions were what would make your trial successful. So you design a protocol that is answering multiple questions. Are you getting enough data? Is your data can be used for next trial? Is it regulator gonna accept that data? All those questions. The protocol was designed based on that. And then comes the compliance. You need to know procedures, you need to implement monitoring system in place, data management system in place. Do you do a paper data or do you do electronic data? All these decisions are important decisions when you're designing a clinical trial. And the strategic factor is that not just the design of the trial or the execution of the trial, but how do you stage your coverage in conferences. So you need to align yourself that I'm gonna have 5 patients and I will use those 5 patients for this conference. Because what you want to do is to share your data with more researchers and doctors who are key opinion leaders, and you wanna just be sharing the results so they can see what you are doing. And we were very honest with our data and we built credibility. We didn't only show good results, right? We showed exactly how we designed the study and what we saw. And Matthew, our scientist, did a great job, very authentic, and the credibility was amazing, like how doctors were talking about how we are presenting. So it's important for any innovations to make sure that you attract people during this process. These are the people become our advocate later, right? So clinical trial, regulatory, I'm not sure if you know this, I thought it would be good to share that every country has a different requirements. So you have FDA, Health Canada, and Europe in our case, and they all have a very specific approval requirements. Either you have an in-house regulatory person who can do it, or you have a consultant who can do it. These are tactical things. These are not strategic things, right? Once you decide what you want to do, you can have people execute that. But very critical part. And the most critical part is that hospitals doing research, they have a very different financial model. So it costs you a lot even in hospitals. So you need to also figure it out, who you want it on your cohort, like which hospitals are brand name important to you or not. If you go after brand name, are you willing to pay for them? And they charge a lot. So we kind of created that balance. We did some high-end, names, and then we did some not so famous hospital because we had to do within the budget that we had. Now comes to regulatory strategy. I mean, we have decided that we're talking about medical device. When I put together, I have had experience when people are developing software and asking if it is a medical device or not, right? So you can have an innovation. These days, there are many software applications and people are like looking at, is it a medical device or is it like a, health, what do you call it? Health management outside of the medical device, right? We are in a medical device, so for medical device company, anything that you develop after prototype is done, not before the prototype is done. After the prototype is done, you need to have a formal documents that regulator wants to see. So the company doesn't need to be ISO certified or anything, but the structure, in having a structure in place allows you to use those documents later for the regulatory submissions. So it's a balance, like it's costly. Some company gets certified and they lose it because they don't have maintenance in place. But for regulatory approval, ISO structure is a must. I would say that formal design and development documents are must, like what your requirements, what specification you used, what kind of a testing did you do, they all need to be a formal documentation. And then comes the validation. So the validation could be bench testing. In our case, it was all three. In some innovations, if you don't need clinical data, bench testing is enough. It depends on what device you are developing and what we need to address through the regulation. And then all that is coming together as a market clearance application. Quality management system requirements are, I mentioned this already, that you don't need to certify your company, but put a structure in place. That structure is very, very helpful down the road. When do you conduct formal clinical validation? If prototype is ready to be manufactured. For us, our first preclinical, we only used one system. For our phase 1 study, we used 3 systems. And for our pivotal study, we used 14 systems. So you can imagine building one system, engineering can do it. You don't need to have a manufacturing. Building 3 systems also, you can have a combination. Somebody can build 3 systems for you. But if you have to build 14 systems consistently, you need to have a structure in place. You need to have some sort of a supplier management in place, so you're buying things that are compatible with your future plans. Because as soon as those 14 systems were done, we went into commercialization mode. That means you are now talking about many more sites that you need to have a product in. So the staging, those are very, very important. Performance data is it's a subjective term, but when you come into the regulatory world, it is not so subjective. It depends on what kind of a technology you have. The performance data will be defined based on that. If you are making it, as I mentioned, if you are making it prototypes, you don't need to worry about that. Your performance data can be used from that device to the next level device to the next level device, as long as you don't make any significant changes. When I say significant changes, in our case, you can imagine what very early picture looked like versus what our commercial picture looks like. Yes, there were tons of changes, but when I say significant changes, it's like a principle of operation cannot change. If you are looking at, you have selected dual frequency algorithm, you just can't change that. If you do, you invalidate your preclinical work. if you are changing power or energy, deposit, you can invalidate your previous work. I'll show you that we made changes, but how we kept regulator engaged, so those changes did not invalidate our previous work. So for evaluating that, what level of performance data I already have and what I need, it is, is this ready to be shared with the regulator or not? So we were sharing bench testing, animal testing, and human testing data with regulator, because each stage, one, you are really creating a proof of concept. The other one, you are creating a safety and feasibility that your technology can be used in vivo. And then you are creating a human data for efficacy. And these regulatory submissions are always going back to what your device is designed for. If your device is designed for specific intended use, the performance has to be for that use. And additionally, marketing claims are important, right? So if in our case, if marketing wanted to say that we treat prostate cancer, now you are looking at a very different study design, right? So we had to say that we ablate a tissue in a patient population that has a low to intermediate risk prostate cancer. So very different claim. So claims are important. More on preclinical work. So you need to know what animal model you are selecting. And it's always a good idea to go to regulator and see if they agree to it. The clinical study design it's developed based on that data, and the primary and secondary endpoint are also, because in preclinical you are using certain parameters that you need to use it in a human, and so it's important to know what that performance data looks like. And the device parameter, there are some you can change. If you are changing, let's the water flow system. As long as the water is flowing the way you said it's flowing, it doesn't matter if you change it from hanging somewhere to somewhere else. They will not care about it. But if you change frequency, as I mentioned, yes, they will question it. How does it impact your manufacturing? What your transducer looks like? What kind of a testing you have done with it? All kind of questions will come. Preclinical testing is like, it doesn't answer all the questions, right? So in a preclinical testing, you must understand the limitations that you have. In our case, it did not address any toxicity concern. It like you have no symptomatic data that what patient will feel, because when you go in patient, you do need to develop a risk-benefit profile. So you need to tell people what are the risks with your device. With animal study, you can't develop that. So you have to anticipate based on literature that the toxicity will be this % or for this particular thing. So you need to pick things that are relevant to your procedure and identify what risk patient will see, anticipated risk patients will see. And then for the going into the human, we had to, I'm keep repeating parameters, but I can't tell you exactly what those parameters are, but keeping that in mind that you are making decisions from animal to human study, and you have to decide what those parameters are that will allow you to get into human. And regulatory studies, so these are all performance. You can ask more questions later if you have specific ones. I made it too general, so I hope it's not too confusing. I will be very happy to answer any specific questions you may have. Our animal model was, so sorry if you didn't hear the question, is what was our animal model and how did we select that? Our animal model was canine. I will show you more on what the challenges we had with that model. And the regulatory strategy had to change based on the model we picked. And there was no other model that we could use. Um, so the regulatory strategy is that we need to decide which market, which country you want to go first. If there are similar devices in the market, they call predicate. If they are, then you have to follow what they have established. You have to probably do the same. If they're not, then you have to come up with your own criteria. In our case, we had to come up with our own criteria. You have to know what risk class. Based on the risk class, your study design changes or the performance data changes. And then we have to know what standards we need to comply with. And that's a very specific regulatory assessment that has to happen early during the development stages. So engineers are designed to comply with that standard. So in our case, you have electrical, like EMC, electromagnetic, and MR standards. They were obvious one. We had a biological, like a biocompatibility standard, sterility standard. So all these standards are depends on what you're designing and when do you need to comply with it. And earlier you do during the development stages, better it is, cheaper it is. If you have to redesign because someone missed that, and at the end regulatory said, well, you don't have this compliance report, at that time it's going to cost you a lot of money and time. So having that early on is really important. Yes.
What was your US in the US?
Sorry, I can't hear you.
What was your device in the US? Was
I'm not able to follow you. Do you have a mic for them to speak to?
What was the device class in the US?
Device, yeah, I'll show you. I have that in a next slide. Sorry, I just distance, I could not hear you. Yeah, so your strategy cannot be standalone ever. Always have right members involved in your strategy. I mentioned engineering and finance and clinical, but don't forget subject matter experts. For example, when we were developing this design at that time, safety standards were changing, and the safety standards were going to third edition, IEC third edition. I don't know if you're familiar with that. And during that time, we brought in the safety lab people to say that the new standard is coming in. We are in a design phase. We want to know how do we incorporate that changes now. So because it was not published standard, and we knew it is coming and it's going to come with a deadline, it was a smart thing to do to bring them in-house. They charged us $3,000 for a day, worth it. They sat down, they looked at our design, they developed the strategy with us. So it's not always clinical, regulatory, and engineering. You have subject matter experts that will help you in a process. All you have to know is that be aware of what's changing outside, what applies to you, and who can help you in advance, so you are saving time and money because you don't have to redo it. And regulatory authorities, I can't tell you how many times I talk to my friends who are in regulatory and they don't want to go to FDA. They don't want to go to regulators in general because general sense is that once you go there, you are stuck with their answer. If you talk to them, they will give you the most stringent path and you have to just follow at that time, right? Once you talk to them, you can't back off. It's not true. I have done 6 pre-submission studies for TULSA, pre-submission meetings for TULSA. And I can tell you that it did wonderful things for Profound. One, they saw my credibility face-to-face is like nothing like that. They worked with us to solve problems. They were guiding us ahead of time before we did any study. So for me, Profound saved a lot of money in not redoing things. For FDA clearance, it's a one shot. We got 510K within 90 days. People get stuck in that process for years. Right? So in my mind, that if you have to be strategic in your thinking. You can't just go and start talking everything that you have in mind. You need to plan. You need to be there, um, and walk them through, teach them what you are doing, have them understand what your concerns are, and be very specific with them. It's a wonderful thing to do. They will help you and they will trust you that you're doing the right thing. And then whatever stage you are in, share the information with them and they will be happy to see it. So if this is the question, you're, yeah, so it's important to know what your device classification is. So for us, device classification for TULSA was class 3. And my very first meeting with FDA was that you have to do PMA. And that evolved, that changed, and it changed because clinicians that are supporting FDA, and one of the clinicians was also on our advisory board, clinical advisory board, and they realized that the study design they are asking for prostate cancer, clinician will not accept anything under 10 years follow-up. And these small companies will not be able to fund their trials for 10 years to get the device approved. And they changed it from class 3 to de novo. We had a lot of discussion on, like, we were with other 2 companies, we were, you know, and like for us, we were the only one, because right now this is the only device in US market, but it is 510K, not PMA. So it's a class 2 device, not a class 3 device. For Canada, it is still class 3 device, and for Europe is class 2B device. It's a high risk device. So I'm just talking about pre-submission. In the US, it's called Q-submission. In Canada, it's called pre-submission. You need to decide, do you talk to one or do you talk to everyone, like all country that you are interested in. You have to decide how you state these meetings. You know, you need to decide that, do I wait until at the end before I submit 510K, or do I visit them multiple times, and why? The regulators, they are, their guidance, they will tell you in advance that we have draft guidance, consider this. Because as you're developing something, and if there is something in works, they want you to be aware of it. And it was really helpful to know that draft guidance are coming in fact, and at some point we need to comply with it. If you close your eye from that, you can choose not to comply with draft guidance. Many people talk about that. It is not published, we don't need to worry about it. It's short-sighted if you do that. So I would say regulator can help you with that. They tell you what they're working on and what is coming. So you can prepare yourself for that. And it's not just the, strategic part of this meeting, but there are, you know, number of skills, and I would say soft skills are way more important here. So you need to be very, very solid in your science knowledge. So you don't just go to meet FDA with your CEO, you bring your scientist with you. The risk with scientists is they get into so much detail that derails the meeting. So I love my scientist, but I had to coach him to say, we stick to this topic and this is it. You don't go all over the map here, because they give you limited time and they give you written response. So you take advantage of that. You really need to understand what they do with your information when you give them the information. So landscape is important. You need to understand how it is going to be used in clinical environment. So you can, you know, educate them so they understand the risk of the device. Asking the right question is no brainer, but soft skill, it's like, it's so much. I can talk half a day on this, on soft skills. It's not much talked about subject, but it's a deal breaker. If you really don't understand I have seen people rolling their eyes in front of the FDA. I have seen people saying wrong things to FDA and getting emotional about things. It's it's when you are really believing in something, that passion is understandable, but same time, it's also important that they don't care. They are there for different reasons. Understand them more than try to make them understand yourself. So have two people working towards one common goal. It's like what you want and what they want from you. It has to be mutually agreed on. So soft skills are very, very important. And someone has to be a decision maker. So you appoint someone who can make decisions, what to do. So this is a good example that I thought would help you to understand. When we did the very first meeting, regulatory wise, there is a product code in FDA, and that product code is very specific to type of technology or treatment. I searched it and I thought that these two product codes apply to us, but they said no. So they actually gave us a new code. They had to create a new regulation number. So regulation number, this is a regulatory work. Someone in regulatory will know what this means. But knowing when you go to FDA, this was my one of the slides to FDA. And we had a very specific top discussion on NRG and MIK, very specific questions on that. And we had a very specific questions on why can I not be de novo? Why do I have to be 510K? Because there is a different path for that and different value for that. Because end of the day, you want to take this to a commercialization, right? So it's important for establishing the right regulation upfront. They created the number. I added here 8764340, and they made it as more generic because they told us, FDA told us that there are many companies coming up with different methods of heat, and we don't want to create regulation for each laser and microwave and ultrasound and all that. We just want to create a one for the ultrasound system for prostate tissue ablation. So this is you had a question on preclinical. So the preclinical study, we did this study, and this is one of the questions we asked that we have the validation, which is a preclinical validation, and we had a simulation work. And can we take this information for the next study? Because they don't review it before you start, until you submit the IDE. So pre-submission meeting, we were getting their acceptance on this. Now in this one, you can see because the pelvic region of canine is so small, and their prostate is like packed in there. For human, it's different. So you have more room, you have a different anatomy. And we, when we submitted our preclinical protocol, we decided that actually FDA told us that you need to have a similar parameter that you wanted in human. And in human, we wanted a boundary of heat 3 millimeters away from prostate. So we chose that for preclinical. We treated one animal, and we actually injured the leg of that animal because the heat went outside and heated the pelvic nerves in a pelvic region. And we made a decision internally to change from 3 millimeter to 6 millimeters. So you are only treating a subvolume of a gland, of prostate gland. And how do you go to FDA and say, I did 6 millimeter, but in human, I want 3 millimeter mulching, right? So we had to create a justification for that. And we used anatomy as a justification. And we used MR thermometry as a justification because they asked us for late cell kill in preclinical. Show us what late cell kill looks like. Because Sunnybrook already did acute study. So acute study shows that your heat in MR, like in MR thermometry and in histology matches. And FDA said, well, we don't know what the late cell kill will be, right? So do a study and follow up for 1 month. So we used that, and we said that if we have a shorter section of prostate heated, you can see how far that cell kill goes. If you heat all the way to the boundary, you have no way to measuring how far the cell kill happens outside of the prostate because there are fat layers there, and you may not be able to justify that, right? And they say, yeah, that makes sense, and they agree to it, right? So sometimes when I'm talking about you have to have the same parameters, yes, but also if you have a good justification and good scientific reason for that, they'll accept that. And we did it for from preclinical to phase 1. So my next slide is telling you that we changed not just the size, the ablation size, but we also changed temperature. Like this would have asked for a new preclinical study, but we didn't. We justified it. How did we justify it? So you show them that with 55 degree is what we used for animal and what we used for first phase 1 data that we had. And 55 degree, we had positive biopsies because most tumors are at the periphery. And because you are not heating all the way to the boundary, you're leaving cancerous tissue there. So we justified that if we increase the temperature from 55 to 57 degree, and we reduce the margin from 3 millimeter to 2 millimeter, we will push the heat outside of the prostate safely. We will not have a damage there. So we created computer simulation data for that, and they accepted that. So at no stage, the way we staged the study, that FDA said, you have invalidated your previous work, go back and do that again. None, zero. And that's I'm very proud of. And that is not regulatory. That is absolutely scientific way of showing and clinical way of showing that it makes sense. It is logical to do. I won't read through this, but these are some questions. I know this will be available to people, but this is what, in summary, we decided that we want this to be used for reimbursement also. So we decided to do randomized controlled trial, and we realized that clinically it's not possible because the way radiation works versus ablation work, the endpoints are not same. So radiation endpoints are like 2 years down the road versus ablation is instant. You know now if I have MR images to show non-perfused volume, why do I need to wait for 2 years? So we were trying to convince them that we can do IMRT as a retrospective analysis. So that means you go back to, you create your follow-up, match your follow-up with existing databases. If they have biopsy, if they have like number of criteria, and if they have biopsy, we can use that data and we can provide our data and we can publish as a comparative study. They didn't agree to it. So not everything that you propose to FDA, they accept. Because there were many uncertainties in getting the data. It's like you're going to different hospitals, different timelines, different population. Not everyone had a follow-up, not everyone had a biopsy result, and you can't pick and choose. If you go retrospective, you have to do everything. Right? So that was not accepted by them, so we did not have that. So overall, I would say that from concept to commercialization, these are the main stages. And if you remember my slide of from 2000 to 2019, I would say that we had taken over at a preclinical stage, and we had a first release of TULSA in 2016. So from 2012 to 2016, I would say, pretty aggressive goal for getting a product approved in Europe. And then we did a 115 patient trial that took us, so we started that in 2017, and we finished that so we had to have 12 months. So we did 115 patients in almost like 16 months, 13 sites, and we had 12 months follow-up, and we got the 510K this year in summer, in May. Sorry, August. So, I mean, it may look like from innovation to commercialization, it's a long time, but if you have the right strategies in place, it is not as long as you think. This is my last slide, but I just want to, as a innovators, you may just think that I have a great idea, it has a great market potential, I have money, I can make it happen. This is something that you must keep in mind, and I live by this. When you are a small company, you are hiring people that has experience in a large company. And when people are working in a large company for a long time, they don't have a startup mindset. So they are mostly, they have a job and they want to do a job and they are just subject matter expert. When you talk about innovation and you talk about, people who have a desire to grow, it's a very different group of people that you must recognize. So when I say know your team, it is very, very critical that you have people working with you that you can see they have a desire to either grow themselves or grow the company. And that's how you get successful. Without that team, you're not going anywhere. Because there are a lot of struggles, challenges that you have, financial challenges, you have scientific challenges, you have clinical challenges, you have all kinds of challenges. All you have is a team that you can rely on, right? A team that can get you to the next level. So I would say that keep that in mind. If you ever open a company and if you wanna grow your team, you need to find people that are coachable. And that concludes, wow, right on time.